Chapter 4, Topic 4.3, the fungi. We're going to be going into more detail about fungi in this section. Specifically, we're going to talk about structure, fungal nutrition, fungal pathogens, reproductive strategies, and spore formation. At the end of this recorded lecture, you should be able to list 2 detrimental and 2 beneficial activities of fungi, from the viewpoint of humans, of course, list some general features of fungal anatomy, differentiate among the terms heterotrophic, saprobe, and parasite. Connect the concepts of fungal hyphae and a mycelium and describe 2 ways in which fungal spores arise. It is time to add more depth to what we learned in Chapter 1. But first, a review. What are the characteristics of fungi? Correct. They are eukaryotes, they have chitin in their cell walls, and they do not photosynthesize. They absorb their nutrients from their environment, and they can be either unicellular or multicellular. The text adds a distinction between mushrooms and the like being multicellular and molds being colonial, but for this class you can stick to our original definition. Yeasts are unicellular, molds, which we will lump mushrooms into, are multicellular. Fungi can be either spherical and unicellular. When they're in this form, we call them a yeast. Or they can form filamentous cells that seem like they go on and on forever. We call these hyphae. Now these hyphae, they can be either septate or non-septate. I'm not going to test you on that in this course. But the hyphae can combine into a mass that we call a mycelium. Generally speaking, the mycelium is visible to the naked eye. Think of mold on a piece of bread. You can't see the individual hyphae, but you can sure see the puffball of the mycelium. One last term, dimorphic. Some fungi can be yeast-like in one environmental situation and mold-like in another environmental situation. Usually, dimorphic fungi are yeast-like at body temperature, mold-like at room temperature. That's all the detail we're going to go into on that one. Let's talk about fungal pathogens. Humans, generally speaking, are remarkably resistant to fungal infections. For primary pathogens, most of them cause only mild to influenza-like diseases that resolve on their own. That would be these things like the San Joaquin Valley fever, Chicago fever, Ohio Valley fever. Unfortunately, patients that are immunocompromised by another pathogen, such as HIV, or disease such as leukemia, or other factors such as malnutrition or age, are susceptible to becoming very ill from primary and opportunistic pathogens. Opportunistic pathogens are those that wouldn't cause disease in an immunocompetent or healthy person, or if they are kept where they belong and in the proper numbers. Let's use vaginal yeast infections as an example. The human vagina after puberty is home to mainly lactobacilli, which keep the numbers of other microbes, including the yeast Candida albicans, down. But if a woman becomes ill with something else, say an E. coli-caused bladder infection, she will generally take an antibiotic. The antibiotic takes out the E. coli in the bladder, but it also decreases the numbers of lactobacilli everywhere else, including in the vagina. The Candida see their chance and take over, which is why it's considered an opportunistic pathogen. They cause disease when they get the opportunity, not just by being in and on your body. I will refer you to this table for the list of fungal infections with their pathogens, and it will be a good idea to study this. Mainly what I want you to know here is that fungi release enzymes into their surroundings to break down food into small enough molecules that they can move them into their cells past the cell wall. The cell wall makes it so they can't engulf or ingest their food in chunks. Most of the time the food is dead, making the fungi a decomposer or a saprobe. If it is alive, the fungus is a parasite. Because of this absorptive nutritional lifestyle, fungi are the kings of enzymes. They can make an enzyme to digest just about anything, from cellulose to immersion oil to jet fuel, to name a few. We humans benefit from this because fungi are the great decomposers of the planet. Well, one of them anyway. They take cellulose, which is inedible to most organisms, including humans, and change it into tasty mushrooms. They add flavor to our cheeses. They make our bread and beer, the food and drink that built the ancient pyramids. They also make our wine. And we are working on ways to use them to clean up our environmental disasters. All hail the noble fungi! I won't make you memorize the different types of spore-forming structures of fungi, but I do want you to know their basic reproductive strategies. All fungi can reproduce asexually. They undergo mitosis and generate clones of themselves, like this yeast cell is doing by producing a bud. Filamentous fungi, or molds, also undergo mitosis and form clones of themselves. Sometimes they undergo mitosis and make spores. Spores are resistant forms of themselves that resist drying, lack of food, heat, and so on. Sound like something else we studied in a previous chapter? Yep, endospores. But fungal spores aren't as tough as endospores. Most fungi are able to reproduce sexually. Sexual reproduction involves the combining of two different genomes to create genetic diversity in the hopes that the genetically different offspring will be better suited to the environment than either parent. Not as exciting a definition as you may have hoped for, but that's it. Two different fungi of the same species will fuse two haploid cells to form a new diploid cell. Eventually the diploid cell will undergo meiosis, creating spores that will go off and form new individuals. Yep, fungi spend most of their lives haploid rather than diploid, like animals. If the terms haploid, diploid, meiosis, and mitosis are gobbledygook to you, I wouldn't worry too much. I'd still suggest looking them up, but we won't be talking about haploid and diploid much in this course. That's it for the fungi. You should now be able to list two detrimental and two beneficial activities of fungi from the viewpoint of humans, list some general features of fungal anatomy, differentiate among the terms heterotrophic, saprobe, and parasite, connect the concepts of fungal hyphae and a mycelium, and describe two ways in which fungal spores arise.